Today we will be talking about how to perform an automatic cell counting procedure using the BIOL TC10 automatic cell counter. We will first go over the materials needed. First, we will need the BIOL TC10 automatic cell counter. We will then need the L10 micropipette with the complementary tips set at 10 microliters. You will also need a 0.5 milliliter sample of your cells in a 0.6 milliliter microcentrifuge tube with an additional 0.6 milliliter microcentrifuge tube that is empty. In order to insert your cell sample inside of the BIOL TC10, you will need counting slides that have a dual chamber for cell counting. Lastly, you will need trypan blue dye that will stain the dead cells blue in order for the cell counter to get an accurate live cell and total cell count. To start the procedure, you will first need to place a sterile pipette tip on your L10 micropipetter. Then, using your L10 micropipetter, you will take 10 microliters of the trypan blue dye. You will then place your 10 microliters of trypan blue dye in the bottom of your 0.6 milliliter microcentrifuge tube that was empty. Next, you will titrate in and out of your microcentrifuge tube that contains your cell sample in order to get the most accurate cell counting. Once you have titrated in and out several times, you will remove 10 microliters of your cell sample and place it into the microcentrifuge tube that already contains your trypan blue dye. To mix those together, you will titrate in and out using your micropipette in order to combine your trypan blue and your cell sample. Once your cell sample and the trypan blue are mixed well, remove 10 microliters of that mixture and add it into one of the chambers of the cell counting slides. You will place your micropipette tip inside the half circle space and eject the liquid into the chamber. Once all of the liquid has been dispensed into the chamber, dispose of your micropipette tip in an appropriate biohazard waste beaker and insert your counting slide into the TC10 automatic cell counter. Once the slide is inserted, it will automatically begin counting the cells. Once the cell count has been completed, the screen will appear and it will show you your total count, live count, and percent of live cells. These numbers will tell you how many live cells you have inside of your flasks, and if you are passaging your cells, it will help you determine how many new flasks you will need in order to allow your cells to grow further. In this example, we were able to use the numbers of total count and live count in order to determine that our cells needed to be passaged into 2 new flasks in order to allow for proper growth.